Hi, everybody. So today we're going to talk about neuroimaging techniques, and specifically the techniques that are most frequently used or have been historically used quite a bit. Again, keep in mind, a lot of these techniques are not exclusively used for neuroimaging. They are used for imaging any type of human tissue. So let's get started. So the first thing I want to point out is the difference between structural and functional techniques. Structural techniques are any type of imaging that is mostly concerned with structure. So it's really just an image or a map of the brain. Whereas functional techniques are more about the activity of the brain or the function of the brain. A lot of times these actually are mapping blood flow to specific areas of the brain. So structure is about imaging and functional is about seeing what's happening within the brain and the blood flow. The first type of technique we're going to talk about is the EEG, or the electroencephalogram. So electro is just for electrical activity, and encephalo means the brain, and gram represents a graph. So basically you're measuring electrical activity of the brain and graphing it. Here's a picture of what it usually looks like when someone is getting an EEG, and usually there's a lot of different electrodes placed on the scalp. And so this is measuring the activity, the electrical activity of the brain. And it's more of a general activity of the brain, so you're not measuring specific areas necessarily. These are going to map what you may have heard of as brain waves. And this will give you an overall idea of what kind of activity is going on in the brain. So these are very sort of, I guess you would call it broad functional techniques. You're kind of looking at function, but it's an overall activity. And it's not very specific. So you might be able to tell if someone is highly alert or if they're asleep or what stage of sleep they're in, but not a lot more than that. The next type of imaging we can talk about is the CT scan, sometimes the CAT scan, but CT stands for computerized tomography. And tomography is used in a couple of the techniques we'll talk about today. And really tomography just refers to taking a cross-section of a 3D object, just like is presented here in this human head. There's sort of a cross-section, and then you're looking at a 2D image of that slice. So on the right, you see what that 2D image looks like in that slice. And tomography goes and takes individual image slices of the brain so that you have many, many, many 2D images, but you can look at them in succession to sort of get an idea of what the 3D structure looks like. Now in a CT, this uses X-rays to obtain these images. And again, here we're really just looking at structure. And the structure here is not as detailed as some of the other techniques. So a CT scan might be used to reveal differences in the size of certain brain areas. A tumor can be seen on a CT scan, or a large area of damage, such as from a stroke, might be seen. The next type of scan is called a PET scan, or a positron emission tomography. Again, you notice the word tomography here, which means we're still taking a 2D image of a three-dimensional object. We're looking at slices. And positron is an antiparticle of an electron. It's something that's emitted in a radioactive material. And in this case, the patient actually ingests the radioactive material. So they can either do this via injection or by mouth, but either way, there's a radioactive substance that moves through the blood of the patient, makes its way to the brain. And the scanner actually detects the level of radioactivity in various levels of the brain, and that indicates how much blood flow is going to those regions of the brain. So the results are taken in by the computer and usually color-coded. And so red might be high activity, blue might be low activity, and this gives the researcher a good idea of the activity in different areas of the brain. Now, this is a good functional technique, but there's not a lot of structural information. So the next technique we'll talk about actually combines the two to get sort of a good idea of both. So the next image is MRI, or magnetic resonance imaging. Now, MRI can be either structural or functional. And sometimes you'll see a little S or a little F in front of MRI to denote it's either structural or functional. And a lot of times these are both done simultaneously, or at least in the same scan. So you get information that is both structural and functional. Now, SMRI, the structural type, produces high quality images, higher quality than the CT scan. And the benefit of this is that the patient does not have to ingest the radioactive material. The way that it looks at these images or creates these images is using magnetic pulses. And these basically move the molecules of the brain, and the computer tracks the energies that are released by that movement. And that creates a picture of the brain. Like I said, it's a much more detailed picture. So if you compare this to the picture on the CT slide, you'll see there's a lot more detail here. You can clearly see the sulci and gyri. You can even see the little crevices on the cerebellum. You can look at gray matter versus white matter. Specifically, if you look like right around the ventricles, you'll see that gray area that's very clearly delineated from the white matter. So a much clearer image. And then at the same time, you can also use this image to map on functional information using the fMRI. Now, the fMRI uses that same MRI technology, but instead of measuring all molecules, it measures specifically hemoglobin. And the book talks about the bold signal. And the bold signal is actually this measure that measures how much hemoglobin is going to certain areas of the brain. Again, measuring technically the activity of certain areas of the brain. And you can map that right over that structural MRI, which makes this a very useful technique. The most recent form of neuroimaging is DTI, or diffusion tensor imaging. And this really is a type of MRI scan. And instead of measuring the movement of molecules as they realign or of hemoglobin, this actually measures the movement of water. And we have a lot of water in our brain, and how it moves through the brain can give us some indication of the actual neurons within the brain. So if you can imagine sort of like a garden hose, when you turn on the water in that garden hose, the water moves through the garden hose. So in the same way, water would move through an axon easier than it would move outside of the axon, so crossing axons. And so when you measure how the water is flowing through the brain, you can get an idea of how the neurons are structured, where the axons lie, how the water moves is basically indicating the path of axons. And so you can use this information to path out bundles of neurons within the brain and get an incredibly detailed image. And obviously this has had a little bit of special effects added to it in the form of the underlying the brain, but the neuron fibers, or what looks like fibers, are actually what's been mapped through the DTI imaging. So hopefully those overviews really helped you understand a few of the different imaging types. We'll be talking about a lot of different types of imaging going through this class. So this should help you have at least some sort of basic understanding about what type of material or what type of information you can get from each of these different types of scans. And I hope to see you guys again soon. Bye.